Everyone, thank you for coming out tonight. My name is Melaina Yender. I'm a senior at the Montrose High School, and today I'll be talking about my internship at the Scripps Research Institute studying neuroscience and click chemistry. I'll begin by describing what my internship was like and all the experiments that I conducted. So as I had said before, my internship involved in neuroscience and click chemistry, specifically clearance-assisted tissue click chemistry. So click chemistry is essentially the process of binding two molecular entities together, and the tissue part of that is the ability to apply that to the tissue and animal cells, and that way you're able to trace things that are binding to the receptors in animal tissue. And then the clearing part of this is getting rid of all the lipids or the fats in the cells in order to image the cells in the tissue properly. So this was a pretty lengthy process, but after clearing all of the tissues and all of the cells, you were able to attach a fluorescent tag to a molecule that is in the brain tissue that you have. And then I was able to image using a certain kind of microscopy that excites the fluorescent tag, and you were able to see where the drug went. And the applications I had, as I kind of stated, was I traced hallucinogenic drugs through the brain of mice models. So this was truly incredible because using Catch, I was able to see where specific drugs went to in the brain and what receptors and neurons that they binded to. It was truly incredible to be able to see this and understand how the drugs work. The importance of this is being able to know where they go, because understanding the specificity and the accuracy of this drug, or many different kinds of drugs, is able to help scientists be able to learn how this works and what causes specific effects, and then the unknown and adverse side effects that they do not want. And that comes from the drug being very inaccurate. Being able to understand this leads to the modification of the drugs and being able to get rid of adverse side effects. And this is hoping to be expanded into pharmaceutical drugs, so that way there's no side effects with any medication. So I conducted several different kinds of experiments, and I'll try to briefly explain them. The primary was Catch experiments. I was applying Catch to different kinds of drug probes, conditions, and then I was able to apply that to the electrochemistry experiment, which was trying to get the drug to permanently bind to the neuron receptor, so that way any kind of drugs can be used to study. And then this was used through electricity, which was very interesting. And then Catch was also tried to volumetric samples instead of very thin slices. So beginning with the Catch process, it was a pretty lengthy process. We dealt with 100 micron slices of brain tissue. This is very thin. It is the thickness of paper. So it was very, very delicate to work with. And it took about a week in order to go from injection all the way to being able to image the tissue. So we begin by injecting and perfusing and harvesting the brain. Then we would take the tissue here and then run it through the vibratome, which is what would slice the tissue. Then from there you go through the clearing process, which would get rid of all of the fats, and then polymerize and mesh in order to keep all of the cells and cell organelles in place. Then they would go through incubation, where the fluorescent tag would be attached to the drug into the tissue, and then after that we would be able to mount them on the slide, image it, and see where the drug was in the brain. The specific experiments I did was studying the different probes, so I dealt with a lot of different drugs, about 9 or 10 of them. The primarily ones I dealt with was ketamine and partoline. Then I also worked with pre-blocking experiments, which is testing to see if the probe that was chemically made for us specifically worked just like the parental drug. And I also was a big part of modifying the process of Catch and being able to make the incubation buffer more efficient and not use as many reagents, which is something Then the primary thing that I was really involved in besides that was the electrochemistry experiment. So this was something that they would just start to develop as I first got there. It is trying to change the binding of the drug in the brain and into the tissue using electricity. So electrically activating it would make the drug not detach from the neuron, and any drugs can be studied, because this is very limited to what kind of drugs can be studied, because it takes so long and you don't want the drug to wear off. So the process was applying it to lysates, which was homogenized tissue, and running that through a gel, which essentially pulled the proteins down, and then we would be able to see which proteins that the drug had attached to, and study it through there and see if this would actually work, and then being able to apply it to a live mouse when it's injected, and then have it long-lasting in the brain. And then the other experiment I was involved with was volumetric experiments, and being able to apply it to full organs. So we worked with hemispheres of the brain and then also lung samples. So my mentors had a grant to study lung cancer drugs through Catch, and being able to see if lung cancer drugs actually work. So that was also a very credible experience for me. There were a lot of surprises about my internship. There was a lot with my internship that was unexpected. I wasn't expecting the workload, so I did about 2 to 5 experiments every single day, and most of the time they were not related. It was definitely a lot, but I definitely enjoyed it. And then the specifics of them, they were very different. I only expected to do the first slide that I showed you guys, not anything else. So that was definitely something that I was not expecting. And then working directly with the mice, I did not know that I'd be working with the mice until my mentor brought a cage of mice on the 4th day saying, prepare the injection. I'm like, okay, we're gonna do this. But yes. Then one of the biggest surprises about my internship was that by the end of this year, I will hopefully have authorship of the paper for the electrochemistry experiment that I did, which was incredible. Thank you. Yes. Wonderful. Okay, so now I'm done talking about my internship, and now I can talk about what I did in my spare time and on the weekends. So being in San Diego was an incredible experience for me. I've never been to California before, let alone an ocean. So this was truly an amazing experience to be able to experience this. And I was in an Airbnb with 4 other interns and a host mom, and it was truly incredible to be able to experience this. We went to many different places, many different beaches. It really added to the whole experience of being there. Okay, I also wanted to show pictures of the lab trip that we went on while we were there. On the very last week, we went fishing in Point Loma, saw a lot of dolphins, and had a lovely time with my lab. And then finally, advice for future Pinterns and those who are wanting to apply to this program. Biggest thing I would recommend is contacting your mentor and being able to have a connection with them beforehand, so that way you know what you're getting into, as well as afterwards. So I have contact with my mentors and people in my lab currently, so I was offered a job back at the lab as a paid research assistant, and I also have letters of recommendations from people there as well. So this is an amazing connection that you can have to the people to help you with your future for a very long time. Always be open to any opportunity. You don't know what's going to happen out of this. You can't really expect what's to come. So be open and take advantage of all of the information that you're given. You're here to learn. It's going to be a once-in-a-lifetime experience, especially at this age. So learn everything that you can, because it's going to be incredible. And thank you to everyone for my presentation. My internship. And thank you for listening to my presentation.